Okay, let's unpack this. Have you ever, really considered the invisible architectures holding your cells together? Or maybe the silent messengers orchestrating some of your body's most fundamental processes?
Hmm. Things like just breathing, or even how our blood clots.
Exactly. Well, today we are embarking on a deep dive into some of the, well, often unsung heroes of biology.
Hmm.
We're talking about a crucial group of lipids, phospholipids, glycosphingolipids, and eicosanoids.
A really fascinating world.
It is, and our mission today is to sift through a pretty rich collection of sources, articles, research papers, our notes, and pull out the most vital nuggets of knowledge, basically giving you a comprehensive shortcut to truly master this info.
Kind of like getting the elite professional's view on it.
That's the idea. Get ready for, I think, some surprising facts and hopefully plenty of aha moments as we uncover just how indispensable these molecules are.
Yeah, and what's truly captivating here is that these aren't, just inert building blocks. They're not simple fats just sitting there.
Right.
They are dynamic, active players. In, well, virtually every aspect of your cellular life, they're critical for everything from forming the basic structure of your cell membranes, literally defining inside versus outside a cell.
The fundamental boundary.
Precisely. All the way to dictating these complex, nuanced signaling pathways that let your body respond instantly to its environment, fighting an infection, healing a wound, that kind of thing.
Okay.
So we'll explore how they connect, what exactly happens when these, delicate pathways get disrupted.
Which leads to disease, often.
Often, yes. And how our understanding of them directly impacts, well, both diagnostics and developing new therapeutics. You'll really see how interconnected these tiny molecules are to your overall health.
All right, let's start our journey then, laying the foundation with phospholipids. You mentioned they're the predominant lipids of cell membranes. So what is it about their core structure? What makes them so suited for building something so critical?
Well, at their core, phospholipids are these remarkable polar ionic compounds. You can almost think of them as having this clever split personality.
Okay.
The term is amphipathic. It just means each molecule has two distinct parts. First, there's a hydrophilic head, that's the phosphate group, plus an attached alcohol like serine or ethanolamine or choline. This part loves water.
Hydrophilic water-loving. Got it.
Exactly. Then second, there's a long hydrophobic tail, usually fatty acids or similar hydrocarbons. And as the name suggests, these tails are water-fearing. They actively shy away from watery environments.
Okay, so a head that likes water, tails that hate it, and the backbone holding it together.
The backbone could be either diacylglycerol DAG for short, or sphingosine. And that phosphate group is a key part of that water-loving head.
So this dual nature, one part loving water, the other hating it, that's the key. How does that split personality let them form something like a cell membrane?
Precisely. That amphipathic nature isn't just some quirky feature. It's absolutely crucial for their main job. Spontaneously forming cell membranes. If you picture the cell membrane, maybe like a figure 17.1a, it's a double layer, right? A bilayer. The hydrophobic tails, avoiding water, they tuck inward. They associate with each other and other nonpolar stuff like cholesterol, all hidden away inside the membrane, safe from the water.
Okay, tails inward.
Right. Meanwhile, the hydrophilic heads, the water-loving parts, they extend outward on both sides of the membrane. They're constantly interacting with the watery environment inside the cell and outside the cell.
Ah, I see. So heads face the water, tails hide from it.
Exactly. And this clever setup creates an incredibly stable yet still fluid barrier. It's what controls precisely what gets into and out of the cell.
So they're everywhere in cell membranes, literally defining the boundaries of life. But you hinted they do more. What else do these versatile molecules do for us, beyond just structure?
Oh, absolutely. Their functions go way beyond just forming membranes. Even within the membrane itself, they act as critical reservoirs for intracellular messengers.
Reservoirs, like storage tanks.
Sort of. Think of them as tiny loaded springboards, ready to be cleaved, cut by an enzyme, to instantly launch vital signals deeper into the cell. And for some proteins, phospholipids actually act as anchors, tethering them right to the membrane.
Okay, messengers and anchors. What about outside of membranes?
Right, non-membrane roles too. Take your lungs. Phospholipids are key components of lung surfactants.
Surfactant, right, critical for breathing.
Absolutely critical. Without it, the alveoli, those tiny air sacs where gas exchange happens, they'd collapse. And then in your digestive system, they're essential in bile.
Bile, how so?
Their detergent-like properties are vital for solubilizing cholesterol. helps prevent gallstones, helps digest dietary fats. It's really quite remarkable how their structure lets them do such a different critical jobs all over the body.
It really is. Okay, so with that foundation, phospholipids as gatekeepers, signalers, surfactants, detergents, let's delve a bit deeper. Because, like you said, it gets complex. They come in different families.
Indeed, we classify them into two main classes based on their backbone. Some have glycerol, others have sphingosine.
Glycerol or sphingosine, okay?
And it's important to remember, both classes aren't just structural bits, they're active participants, generating lipid signaling molecules too. Very dynamic.
Got it. So the first class, glycerol-based.
Right. Those are the glycerophospholipids, sometimes called phosphoglycerides.
Uh-huh.
So the major class, the vast majority of phospholipids in our membranes are these guys. And they're all derivatives of or contain something called phosphatidic acid, or PA.
PA, phosphatidic acid.
Yes. If you look at a diagram, like figure 17.1b, you see PA is basically diacylglycerol, DAG, with a phosphate group stuck on carbon 3. It's the simplest one, and it's the precursor for all the others in this group.
The starting point.
Exactly. The magic happens when that phosphate group on PA gets esterified, chemically linked to an alcohol-containing compound, like serine, choline, etc. That's how you get the whole diverse family of glycerophospholipids.
Okay. Now, within that family, one name really stood out, cardiolipin. It sounds intriguing. Where does this secret agent operate, and why is it so vital?
Cardiolipin, or diphosphatidylglycerol, is structurally quite unique. Imagine taking two PA molecules and linking them through their phosphate groups using another glycerol molecule in the middle.
So like a double phospholipid?
Pretty much, yeah. You can see it in figure 17.2. But what's really fascinating and medically important is where it lives. While it's common in bacteria, in us eukaryotes, cardiolipin is found almost exclusively in one place, the inner mitochondrial membrane.
The powerhouse of the cell, inside the mitochondria.
Exactly. And its job there is absolutely critical. It maintains the precise structure and function of certain respiratory complexes in the electron transport chain.
Which is essential for making ATP, our energy currency.
Absolutely essential. Which then raises this really interesting question. Why would a molecule so vital for our energy production be recognized by antibodies against a bacterium? Specifically, Treponema pallidum, the syphilis bacterium.
Wait, what? Antibodies against syphilis recognize cardiolipin?
Yes. The old Wassermann test for syphilis actually relied on finding these antibodies. It's a compelling hint at, well, maybe deep evolutionary connections, or perhaps molecular mimicry, where our molecule just happens to look like a bacterial one. Really makes you think.
Wow. Okay, that is fascinating. Moving on from cardiolipin, you mentioned other unique variations like plasmalogens. They have a chemical tweak.
They do. Plasmalogens are a special type of ether phosphoglyceride. The key difference is at carbon 1 of the glycerol backbone. Instead of the usual fatty acid attached by an ester link, it's replaced by an unsaturated alkyl group attached by an ether linkage. This ether bond is actually stronger, more resistant to oxidation than an ester bond.
Ah, okay. An ether link instead of an ester link at C1. And where do we find these?
They're quite abundant in mammals. For instance, phosphatidylethanolamin, you can see its structure in figure 17.3a, is really common in nerve tissue. It's thought to help protect against oxidative stress there. Another one, phosphatidylcholine, is abundant in heart muscle.
And you can spot them by the name AL instead of UL.
Often, yes. That's a good clue. Phosphatidylethanolamin, for example.
Okay. Now, let's switch to a molecule that packs a huge punch, even in tiny amounts. Platelet activating factor, PAF. Why is this one such a heavyweight?
PAF is another ether glycerophospholipid, but it's got a unique twist that gives it immense power. Like plasmalogens, it has a saturated alkyl group at carbon 1 with an ether link. But at carbon 2,
Yes.
instead of a long fatty acid, it has a very short acetyl residue, just 2 carbons. You can see this compact structure in figure 17.3b.
So a short acetyl group at C2 instead of a fatty acid, that makes it so potent.
Incredibly potent. It's one of the most powerful bioactive molecules known. It can cause significant effects at concentrations as low as 10-11 molar, just vanishingly small amounts.
Wow. Like a tiny alarm signal setting off a huge response.
Exactly. It's made and released rapidly by lots of different cell types, acting as this critical rapid response signal. When it binds to its receptors, it triggers really potent thrombotic, clot-forming, and acute inflammatory events.
Inflammation and clotting.
Yes. It activates inflammatory cells, mediates hypersensitivity, inflammation, even anaphylactic reactions. It causes platelets to aggregate and activate key for clotting. It also tells immune cells like neutrophils to make superoxide radicals to kill bacteria. and systemically, it lowers blood pressure.
So a tiny molecule with a massive widespread impact. Truly a heavyweight.
Definitely.
All right, let's shift gears now to the other backbone we mentioned, sphingosine. And the key player here, you said, is sphingomyelin, the myelin mastermind.
That's a good name for it. So instead of glycerol, sphingomyelin uses the amino alcohol sphingosine as its backbone. Sphingosine itself is built from serine and palmitate. Figure 17.4 shows its structure.
Okay, sphingosine backbone. How is sphingomyelin formed?
It's a neat process. First, a long-chain fatty acid, LCFA, attaches to the amino group of sphingosine via an amide linkage. This creates a molecule called ceramide.
Ceramide? I think you mentioned that's a precursor for other things, too.
Exactly. Ceramide is a really important intermediate. It's the precursor for sphingomyelin, and all the glycolipids we'll get to later.
Okay, so ceramide is formed. Then what?
Then, the alcohol group at carbon 1 of the sphingosine gets esterified, linked via an ester bond to phosphorylcholine, usually donated from phosphatidylcholine. The result is sphingomyelin.
Got it. Ceramide plus phosphorylcholine equals sphingomyelin.
Essentially, yes. And it's critical to know, sphingomyelin is the only significant sphingophospholipid in humans. Its main claim to fame is being a major part of the myelin sheath around nerve fibers.
The insulation for our nerves.
Precisely. It insulates and protects the neuronal axons in our central nervous system, ensuring nerve impulses travel quickly and efficiently. Without good myelin, you get serious neurological problems.
Makes sense. Okay, we understand what these molecules are. Now, how does our body actually make them? You mentioned it's like a sophisticated assembly line.
It really is. The core idea, especially for glycosphingolipid synthesis, involves activation. Either the DAG backbone or the alcohol head group that needs to be added gets activated.
Activated how?
By linking it to cytidine diphosphate, CDP. So you get CDP-DDAG or CDP alcohol. This makes the subsequent reaction, joining the two parts together, energetically favorable. CMP gets released as a byproduct.
Similar to how sugars are activated with UDP.
Exactly the same principle. It's a common strategy in biosynthesis. And another layer of complexity is the fatty acids used. Typically, you get a saturated one at C1 and an unsaturated one at C2, but there's variation, which adds to the diversity of phospholipids.
And where does this assembly happen?
Most of it takes place in the smooth endoplasmic reticulum, the SER. From there, the newly made phospholipids are transported to the Golgi, then shipped out to other membranes or even secreted from the cell.
Any exceptions?
Yes, ether lipids, like the plasmalogens we talked about, actually start their synthesis journey in peroxisomes. Shows the intricate organization within the cell.
Okay. And what about the starting point for glycerophospholipids, that phosphatidic acid, PA?
Right, PA. As we said, it's the precursor for all other glycerophospholipids and also for triacylglycerol, TAG, our main fat storage molecule. PA itself is made from glycerol 3-phosphate and 2 fatty acyl-CoA molecules.
And most cells can make phospholipids.
Almost all of them, yeah, except mature red blood cells, which lack the machinery. But TAG synthesis, that's much more restricted, mainly liver, adipose tissue, lactating mammary glands, intestinal cells. Shows how fundamental phospholipids are for all cells.
Now, phosphatidylcholine, PC, and phosphatidylethanolamine, PE, you said they're the most abundant ones. How does the body make sure it has enough?
Well, the main route uses choline and ethanolamine that we get either from our diet or by recycling existing phospholipids. It's pretty straightforward.
How does that work?
First, the choline or ethanolamine gets phosphorylated by a kinase. Then it's converted into an activated form, CDP choline or CDP ethanolamine. Finally, that activated head group is transferred onto DAG, releasing CMP. Figure 17.5 shows this pathway clearly.
Okay, so phosphorylation, activation with CDP, then transfer it to DAG.
Exactly. And here's a really critical point about choline. While our bodies can make some, we don't make enough to meet our needs.
So we need it from our diet.
Absolutely. Choline is an essential dietary nutrient. We need around 550 milligrams for men, 425 milligrams for women daily. And it's not just for phospholipids, it's also crucial for making acetylcholine, the neurotransmitter.
Ah, right. Muscle contraction, memory.
Exactly. So getting enough choline is vital for membranes and nerve function.
Okay, now for that aha moment, you mentioned phosphatidylcholine in lung surfactant, critical for breathing.
Absolutely critical. We're talking specifically about dipalmitoyl phosphatidylcholine, DPPC, or dipalmitoyl lecithin. The key here is dipalmitoyl. It has palmitate, a saturated fatty acid at both position 1 and 2 on the glycerol.
2 saturated tails. Why is that important?
That structure is perfect for its job. DPCC is made by special cells in the lungs, type 2 pneumocytes, and it's the main lipid component, like 90% of lung surfactant. The surfactant lines the alveoli, the tiny air sacs.
And its job?
Its main job is to dramatically reduce the surface tension of the fluid layer lining the alveoli. This makes it much easier to reinflate them after you breathe out. It prevents them from collapsing, a condition called atelectasis.
So without DPPC, breathing would be incredibly difficult.
A monumental struggle, yes. Which leads us to fetal lung maturity.
Right. How do doctors check if a baby's lungs are ready?
They can measure the DPPT to sphingomyelin ratio, the LS ratio in the amniotic fluid. A ratio of 2 or more generally means the lungs are mature.
Why that ratio?
Because around 32 weeks of gestation, the baby's lungs start making much more DPPC and less sphingomyelin. It's a natural biochemical marker of readiness for breathing air.
And if this process isn't ready in time for premature babies?
That leads to respiratory distress syndrome, RDS. It's caused by insufficient surfactant. Tragically, it's a major cause of neonatal deaths. The babies just struggle so hard to breathe.
Is there anything that can be done?
Thankfully, yes. Doctors can give them other glucocorticoids before delivery to help speed up lung maturation in the fetus. And after birth, giving the baby natural or synthetic surfactant directly into the lungs can be life-saving.
That's incredible. Does RDS affect adults too?
It can, yes. Acute RDS can happen at any age, usually after severe lung damage from infection or injury. Fluid builds up, blocking gas exchange.
Okay. Now, back to PC synthesis. You mentioned the liver has a backup plan if choline is low.
It does. The liver exports a lot of PC in bile and lipoproteins, so it needs a steady supply. If choline is scarce, it can actually make PC starting from phosphatidylserine, PS.
How does that work?
First, PS gets decarboxylated to PE, phosphatidylethanolamin. Then PE undergoes 3 methylation steps, adding methyl groups one by one, using S-adenosylmethionine as the donor to become PC. Figure 17.6 shows this pathway.
Wow, quite the workaround. Shows how important PC is.
It really does. The liver makes sure it gets made, one way or another.
Okay, what about phosphatidylserine, PS itself? How is it made?
PS is mainly made via the base exchange reaction. The ethanolamine head group of PE is swapped out for free serine. Again, you can see this in figure 17.6. Its main job is membrane synthesis.
And it has a negative charge.
Yes, a net negative charge, which is important for its interactions, especially as we mentioned briefly in blood clotting, where it provides a surface for clotting factors to assemble.
All right. Now, phosphatidylinositol, PI. You call this one a signaling hub. What makes it so special?
PI is synthesized from free inositol and CDPP-DE, similar to the others, figure 17.5. But what's really key is its fatty acid composition. Very often, it has stearic acid at C1 and, crucially, arachidonic acid at C2.
Arachidonic acid. That sounds important.
It's hugely important because PI serves as a major reservoir of arachidonic acid in membranes. Arachidonic acid is the precursor for prostaglandins and other eicosanoids, those potent local hormones we'll get to.
Ah, okay, so PI holds onto arachidonic acid until it's needed for signaling.
Exactly. Like PS, PI also has a net negative charge. And interestingly, both PS and PI are found mainly on the inner leaflet of the cell membrane, facing the cytoplasm. This asymmetry is actively maintained by enzymes called flippases and floppases.
Inner leaflet. Okay, so how does PI act as a signaling hub?
Well, membrane-bound PI can be phosphorylated by kinases to produce polyphosphonosides. A key one is PIP2, phosphatidylinositol 4,7,5-bisphosphate. You can see its structure in figure 17.7.
PIP2, okay.
Now, the critical step happens when PIP2 gets cleaved by an enzyme called phospholipase C, PLC. This is triggered when signals like hormones or neurotransmitters bind to G protein-coupled receptors, GPCRs, on the cell surface, activating PLC, as shown in figure 17.8.
So a signal outside triggers PLC to cut PIP2 inside.
Precisely. And that cleavage releases 2 vital second messengers, inositol 1000-4455, IP3, and diacylglycerol, DAG.
IP3 and DAG, what do they do?
They work together. IP3 triggers the release of calcium from intracellular stores. DI activates protein kinase C. This combination calcium release and protein kinase C activation sets off specific cellular responses. It's how cells hear and respond to external signals.
Wow. A tiny lipid gets cut, and suddenly the cell takes action.
It's a fundamental signaling pathway. But PI does even more. It can also anchor proteins to the membrane.
Anchor proteins? How?
Certain proteins can be covalently attached via a carbohydrate bridge to membrane-bound PI. This forms a glycosylphosphatidylinositol, GPI, anchor. Figure 17.9 shows this.
A GPI anchor. Any examples?
Lipoprotein lipase, The enzyme that breaks down fats and lipoproteins is attached to capillary walls by the GPI anchor.
What's the advantage of this type of anchor?
It gives the protein more freedom to move laterally along the membrane surface compared to proteins embedded within the membrane. And these anchors can be cleaved by phospholipase C, allowing for regulated release of the protein.
Is this clinically relevant?
Very much so. A deficiency in making GPI anchors in blood cell precursors leads to a serious hemolytic disease called paroxysmal nocturnal hemoglobinuria. Without the GPI-anchored protective proteins, blood cells get destroyed by the complement system.
Yikes. Shows how important even these anchors are. Okay, what about phosphatidylglycerol, PG, and cardiolipin again?
Right, PG is found quite a bit in mitochondrial membranes, and it's the direct precursor for cardiolipin, that double phospholipid specific to mitochondria. PG itself is made from CDT-DAG and glycerol 3-phosphate. Then cardiolipin is formed by transferring a DAG 3-phosphate unit from another CDP-DAG onto a pre-existing PG molecule. It all links back together, especially within the mitochondria.
Okay, let's loop back to sphingomyelin one last time, but focusing on its synthesis. You said it was quite detailed.
It is. It's a multi-step pathway, shown nicely in figure 17.10. It starts with palmitoyl-CoA condensing with the amino acid serine. CoA is lost, CO2 from serine is lost, and this needs vitamin B6, pyridoxal phosphate.
Serine plus palmitoyl-CoA.
Right. The product is then reduced using NADPH to make sphingonine. Then sphingonine gets acylated, a long-chain fatty acid is added, and then desaturated to produce ceramide.
There's ceramide again, the central hub.
Exactly. And ceramides themselves are really important, especially in the skin. They help maintain the water barrier. Low ceramide levels are linked to things like eczema.
Ah, interesting. Okay, so we have ceramide. How do we get to sphingomyelin?
The final step is transferring phosphorylcholine, usually from PC, onto the ceramide. This yields sphingomyelin and leaves DAG behind.
Got it. And you mentioned the fatty acids in sphingomyelin can vary.
Yes, quite significantly. Myelin sheath sphingomyelin tends to have very long fatty acids like lignoceric and nervonic acid, making it very stable. Gray matter sphingomyelin usually has stearic acid. Structure tailored to function, again.
In critical detail. Okay, so these molecules are built, they do their jobs, but they also need to be broken down, recycled. What happens when that process goes wrong? That's often where disease comes in, right?
That's precisely right. Breakdown or degradation is just as important as synthesis. The glycerophospholipids are broken down by enzymes called phospholipids. These are found everywhere in our tissues, in pancreatic juice for digestion.
But also in toxins and bacteria.
Unfortunately, yes. Some venoms and pathogenic bacteria use phospholipids to damage our cell membranes. Sphingomyelin, on the other hand, is broken down by a specific lysosomal enzyme called sphingomyelinase.
Okay, tell me more about the phospholipids that break down glycerophospholipids.
Well, there are different types, PLA1, PLA2, PLC, PLD, and each one cuts the phospholipid at a specific bond. You can see the different cleavage sites in figure 17.1. If you remove just one fatty acid from C1 or C2, you get a lysophosphoglyceride.
Lysophosphoglyceride.
Right. And these phospholipids aren't just for simple degradation. They play a dual role. They release molecules that act as second messengers, like DH and IP3 from PIP2 cleavage by PLC. They also release substrates for making other messengers, like releasing arachidonic acid via PLA2 for eicosanoid synthesis.
So breakdown is also signaling.
Exactly. And it's also crucial for remodeling. PLA1 and PLA2 can remove specific fatty acids, and then other enzymes can put different ones back on. This is how the body makes sure DPPC for lung surfactant has 2 palmitates, or how it ensures PI has arachidonic acid at C2.
Remodeling, is that clinically relevant too?
Yes. Barth syndrome, that rare X-linked disorder with heart problems and muscle weakness, is caused by defects in remodeling cardiolipin in the mitochondria. Shows how vital even tweaking the fatty acids is.
Wow. Okay, now on to sphingomyelin breakdown. This is where we see those genetic disorders, right?
Yes, absolutely. Sphingomyelin is degraded by the lysosomal enzyme sphingomyelinase. It cuts off the phosphorylcholine head group, leaving ceramide. Then another enzyme, ceramidase, splits the ceramide into sphingosine and a free fatty acid. Figure 17.12 shows this.
And the ceramide and sphingosine released, they do things too.
They do. They're not just waste products. They act as signaling molecules themselves, influencing things like protein kinase C activity and promoting apoptosis or programmed cell death.
Okay, so what happens if sphingomyelinase is deficient?
That leads to Niemann-Pick disease, types A and B. It's an autosomal recessive disorder. The enzyme is missing or faulty, so sphingomyelin builds up in lysosomes.
Type A is the severe one.
Yes, type A is the severe infantile form. Very low enzyme activity, less than 1%. Sphingomyelin accumulates massively in the liver and spleen, making them huge. You see these lipid-laden cells, like in figure 17.13. Tragically, there's rapid progressive neurodegeneration because it also builds up in the CNS. Death usually occurs in early childhood. It's more common in the Ashkenazi Jewish population.
And type B.
Type B is less severe. Patients have some residual enzyme activity, maybe up to 10%. Onset is later, survival is much longer, often into adulthood. Neural tissue is usually spared, but lungs, spleen, liver, bone marrow are affected, causing chronic problems. The difference really highlights how even a little bit of enzyme activity can make a huge difference. It's a stark example of lysosomal storage disease.
A tragic illustration of breakdown gone wrong. Okay, from phospholipids, let's move to their relatives, the glycosphingolipids. These bring carbohydrates into the mix, right? Adding another layer of identity.
That's a perfect description. Glycolipids contain both carbohydrate and lipid parts. And since they are derivatives of ceramides, just like sphingomyelin was initially, they're more precisely called glycosphingolipids.
So ceramide is the precursor for both phosphorylated ones like sphingomyelin and glycosylated ones like these.
Exactly. Ceramides are at that crucial branch point. Like phospholipids, glycosphingolipids are essential membrane components found everywhere, but especially abundant in nerve tissue. And crucially, they are located exclusively in the outer leaflet of the plasma membrane.
Outer leaflet only, facing outwards.
Yes, perfectly positioned to interact with the extracellular world. This location dictates their functions. They're involved in cell-cell interactions, adhesion, recognition, controlling growth and development.
And they form lipid rafts.
They do. They cluster with cholesterol and GPI-anchored proteins to form these lipid rafts, dynamic microdomains that organize signaling and trafficking on the cell surface, like little organizing centers.
And they have antigenic properties,
Yeah.
like blood types.
Exactly. Glycosphingolipids are the source of our ABO blood group antigens. It's the specific carbohydrate structure that determines your blood type. They also act as embryonic antigens, some tumor antigens. And remember, it's the carbohydrate portion that the immune system recognizes.
Fascinating. So the sugars are like ID tags.
They really are. Unfortunately, they can also be co-opted as receptors by things like cholera and tetanus toxins or certain viruses, using them to get into cells.
Oh, clever but nasty.
And like with sphingomyelin, genetic defects in their breakdown lead to lysosomal storage diseases. Also, subtle changes in their carbohydrate structures are often seen in cancer cells.
Okay, so how do these carbohydrate additions define their specific identities? What's the structural basis?
It's all about the sugars. Unlike sphingomyelin, glycosphingolipids have no phosphate. The polar head group is a sugar, either a single monosaccharide or a chain of them, an oligosaccharide attached directly to the ceramide by an O-glycosidic bond. Figure 17.12 shows this. The type, number, and sequence of sugars determine the specific glycosphingolipid.
Okay, so different types based on the sugars.
Right. We can broadly divide them into neutral and acidic ones. The neutral glycosphingolipids are uncharged. The simplest are the cerebrosides, just one sugar attached to ceramide.
Like,
Galactocerebroside has galactose, and it's the most common one in myelin. Figure 17.12 shows it. Glucocerebroside has glucose, and it's an important intermediate for making more complex ones. They're mainly found in brain and nerves, especially myelin.
and then more complex neutral ones.
Yes, you could add more sugars to glucocerebroside to make ceramide oligosaccharides, also called globosides, like adding galactose to make lactosylceramide, and then maybe N-acetylgalactosamine. More sugars, more complexity.
Okay, and the other type, acidic ones.
Right, the acidic glycosphingolipids. These carry a negative charge at physiological pH. The charge comes either from N-acetyl neuramidic acid, NNA, acyalic acid, which you find in gangliosides, or from sulfate groups found in sulfatides.
Gangliosides. Those sound complex and important.
They are the most complex, yes, and very medically relevant. They're found mainly in the ganglion cells of the CNS, especially at nerve endings. Structurally, they're ceramide oligosaccharides with one or more nanomolecules attached.
And there's a special naming system.
There is G for ganglioside, then subscripts like M, D, T, Q for mono, D, tri, or quaternana, plus numbers letters for the sugar sequence. GM2 is shown in figure 17.15. They're medically interesting because several lipid storage diseases involve the buildup of these nanite-containing lipids, as we'll see in figure 17.20.
Okay, and the other acidic type, sulfatides.
Sulfatides are simpler. They're basically galactocerebrosides with a sulfate group added, making them negatively charged, found mainly in the brain and kidneys.
Got it. So again, synthesis and breakdown must be balanced. When that balance tips for glycosphingolipids, it leads to severe disorders.
Absolutely. Balance is key. Synthesis happens mostly in the Golgi. Sugars are added sequentially from UDP sugar donors by specific glycosyltransferases, much like making glycoproteins.
And adding sulfate for sulfatides.
That uses the special sulfate donor PPS, 3-L-sulfadenosine 5-phosphosulfate, shown in figure 17.17. A sulfotransferase adds the sulfate to galactocerboside. PP-phylates and noelnicin, PPS is used elsewhere too, like for making glycosaminoglycans. Figure 17.0 gives a nice overview of sphingolipid synthesis pathways.
Okay, synthesis in the Golgi. What about breakdown?
Breakdown happens in the lysosomes. The glycosphingolipids are internalized, lysosomes fuse, and lysosomal enzymes hydrolytically cleave off the sugars one by one.
Following that lysine first off rule.
Exactly, like taking apart a Lego model in reverse order.
Right.
If any enzyme in that degradation pathway is defective.
You get a lysosomal storage disease.
Precisely. Specifically, a sphingolipidosis.
This is that whole class of genetic disorders where lipids accumulate.
Yes. In a healthy person, synthesis and degradation are balanced. If a specific lysosomal acid hydrolase is faulty, its substrate, a specific sphingolipid, builds up in lysosomes. These are the sphingolipidoses outlined in figure 17.20.
And the consequences are severe, especially in nerve tissue?
Often devastating, yes. Neurologic deterioration, often leading to early death. They share common features.
Like?
Usually, a specific lysosomal enzyme is deficient. Usually, only one specific sphingolipid accumulates. Synthesis is normal. They're progressive. Many are fatal in childhood, but there's huge variability in severity, like Niemann-Pick A versus B,
And genetic causes.
caused by various mutations within a single gene. Most are autosomal recessive, except for Fabry disease, which is X-linked. Incidence is generally low, but some, like Gaucher, Tay-Sachs, and Niemann-Pick, are more common in the Ashkenazi Jewish population. Tay-Sachs also in some other specific groups.
How are they diagnosed and treated?
Diagnosis involves measuring enzyme activity in cells, fibroblasts, leukocytes, or DNA analysis. Looking at tissue under a microscope is also helpful. You see things like shell-like inclusion bodies in Tay-Sachs, or the crumpled tissue paper. Look in Gaucher cells, figure 17.1 time. Prenatal diagnosis is possible.
And treatment.
For a few, like Gaucher and Fabry disease, there's recombinant human enzyme replacement therapy, giving the missing enzyme intravenously. It works, but it's incredibly expensive. For Gaucher, there's also bone marrow transplant, since macrophages are affected, and substrate reduction therapy, using drugs to slow down the synthesis of the accumulating lipid.
The study questions mentioned Fabry disease as an example. Can you tell us more about that one?
Sure. Fabry is the classic X-linked one, caused by deficiency of alpha-galactosidase A. It mainly affects males. Symptoms include burning pain in hands and feet, a specific red-purple skin rash called angiokeratomas, and serious kidney and heart problems later in life. Protein in the urine is a key sign of kidney damage. It's one of the few where enzyme replacement therapy is an option.
A tough disease, but some hope with treatment. Okay, finally, let's move to our third group. The eicosanoids. Highly potent, locally acting messengers.
Exactly. This group includes prostaglandins, PG, thromboxanes, TX, and leukotrienes, LT. The name eicosanoid comes from eicosa, meaning 20, because they're derived from 20 carbon polyunsaturated fatty acids, mainly omega-6 and omega-3.
And they're potent.
Incredibly potent. They trigger a huge range of responses, normal things like inflammation, stomach protection, kidney function, smooth muscle contraction, blood vessel diameter, platelet function, but also involved in pathology like hypersensitivity.
How are they different from regular hormones?
Key differences. They're made in tiny amounts in almost all tissues, not specific glands. They act locally, right where they're made, not transported in blood. They're not stored, made on demand. And they have an extremely short half-life, rapidly broken down, very tight control.
Local, on-demand, short-lived signals.
Exactly. They act via GPCRs on cell surfaces, usually affecting cyclic AMP levels. Figure 17.21 shows some structures.
So where do these powerful local mediators come from? How are prostaglandins and thromboxanes made?
The main precursor for the most common ones in humans, series 2, is arachidonic acid. That 20-carbon omega-6 fatty acid we mentioned earlier, derived from dietary linoleic acid.
And arachidonic acid is stored in membrane phospholipids, like PI.
Exactly, usually a carbon 2. It gets released from the membrane by phospholipase A2, PLA2, when the cell receives a signal like a calcium increase. That release is the key trigger. Figure 17.23 shows this.
PLA2 releases arachidonic acid, then what?
The first step in making PGs and TXs is the oxidative cyclization of free arachidonic acid to form PGH2. This is done by the enzyme PGH2 synthase, also called prostaglandin and diperoxide synthase.
GH2 synthase, what does it do?
It's in the ER membrane and has two activities in one protein. A cyclooxygenase, COX activity that uses oxygen to form a ring, and a peroxidase activity that finishes the job using glutathione.
COX activity, that sounds familiar.
Yes. PGH2 is then converted into various specific PGs and TXs by different downstream syntheses, depending on the cell type. Figure 17.23. PGs have a 5-carbon ring, TXs have a 6-membered oxane ring, figure 17.21.
And there are different COX enzymes, COX1 and COX2.
Exactly. This is a crucial distinction. COX1 is made constitutively. It's always there in most tissues. It does housekeeping jobs, protecting the stomach, maintaining kidney function, normal platelet aggregation.
The baseline COX.
Right. COX2, on the other hand, is inducible. It's made mainly in response to inflammation signals from immune cells. The big increase in PGs from COX2 causes the pain, heat, redness, swelling of inflammation, and the fever of infection.
COX1 for housekeeping, COX2 for inflammation.
That's the general idea, yes.
This distinction is obviously huge for pharmacology. How do drugs target these pathways to reduce inflammation, and what are the challenges?
It's central to anti-inflammatory drug action. Cortisol, the steroid, works upstream by inhibiting PLA2, figure 17.23, stopping arachidonic acid release altogether.
Cuts off the supply.
Right. Then you have the NSAIDs, aspirin, ibuprofen, naproxen, indomethacin.
Yeah.
These directly inhibit the COX enzymes, blocking PGH2 synthesis.
But they block both COX1 and COX2.
Those traditional NSAIDs do, yes. And inhibiting COX1 systemically is what causes the common side effects. Stomach damage, kidney problems, impaired blood clotting. That's the downside.
Aspirin has some unique effects, too.
It does. Besides inhibiting COX, it also triggers the synthesis of anti-inflammatory lipoxins and inflammation-resolvins and protectins, a complex drug.
So to avoid the COX1 side effects, they developed COX2-specific inhibitors, COXIBs.
Exactly. Drugs like celecoxib. The idea was to target the inflammation COX2 while sparing the housekeeping COX1. Reduce inflammation without the stomach issues.
Did it work?
Well, yes and no. They did reduce inflammation with fewer GI side effects, but then concerns arose about increased risk of heart attacks and strokes, likely because they inhibited PGI2 prostacyclin production by COX2 in blood vessels, upsetting the balance with thromboxane, leading to a more pro-clotting state. Some COXIBs were withdrawn. It shows how tricky targeting these finely balanced systems can be.
Wow. A real lesson in unintended consequences. Now, that balance between thromboxanes and prostaglandins in platelets, the aspirin story, can you walk us through that?
Absolutely. It's a beautiful example. Thromboxane A2, TXA2, is made by COX1 in activated platelets. It makes platelets aggregate and constricts blood vessels, promoting clot formation.
TXA2 will pro-clotting.
Right. Prostacyclin PGI2 is made mainly by COX2 in the endothelial cells lining blood vessels. It does the opposite. Inhibits platelet aggregation and dilates blood vessels, impeding clot formation.
PGI2 equals anti-clotting.
Exactly. The balance between TXA2 and PGI2 is what normally limits clots to sites of injury. Now, aspirin, it irreversibly inhibits both COX1 in platelets and COX2 in endothelial cells by acetylating them.
Irreversibly, that's key.
It's the key. Platelets have no nucleus. They can't make new enzymes. So once aspirin knocks out their COX1, it's gone for the life of the platelet. 7-10 days, TXA2 production plummets.
Okay, platelets are permanently inhibited. What about endothelial cells?
Endothelial cells do have nuclei. They can make new COX2 enzyme. So while aspirin inhibits their COX2 initially, they can recover and start making PGI2 again.
So aspirin causes a lasting decrease in pro-clotting TXA2 from platelets, but only a temporary decrease in anti-clotting PGI2 from endothelial cells.
Precisely. This shifts the balance towards anti-clotting. That's why low-dose aspirin helps prevent heart attacks and strokes. It reduces thrombus formation by selectively crippling platelet TXA2 production long-term. A brilliant exploitation of cell biology.
That is genuinely fascinating. Okay, beyond the COX pathway, there's the other route from arachidonic acid involving leukotrienes, key in allergies and asthma.
Yes, the lipoxygenase LOX pathway. Instead of cyclization, LOX enzymes add oxygen to arachidonic acid to form linear hydroperoxy acids. For example, 5-LOX converts arachidonic acid to 5-HPP, figure 17.23, which is then converted into various leukotrienes, LTs.
Leukotrienes, what do they do?
They are potent mediators of allergic responses and inflammation. They cause bronchoconstriction, increase vascular permeability, attract immune cells, major players in asthma and allergies.
So drugs for asthma target this pathway.
Yes, inhibitors of 5-LOX itself and drugs that block leukotriene receptors are common asthma treatments.
How does this pathway interact with inhibitors?
Cortisol inhibits leukotriene synthesis because it blocks PLA2 upstream, cutting off the arachidonic acid supply. But crucially, NSAIDs do not inhibit the LOX pathway. They only block ALOX.
So if you block KOX with an NSAID,
The arachidonic acid that would have gone down the COX pathway can get shunted over to the LOX pathway instead.
leading to more leukotrienes.
Exactly. This explains aspirin-exacerbated respiratory disease, AERD. In some asthmatics, 10%, taking aspirin or other NSAIDs causes a massive overproduction of leukotrienes, leading to severe bronchoconstriction. It's a dangerous shunting effect.
Wow. Another complex interaction. Okay, summing this all up, what's the big picture? We've covered phospholipids, glycosphingolipids, eicosanoids, intricate structures, vital roles.
The big picture is that these aren't just footnotes in a biochemistry textbook. They are fundamental players. Phospholipids building membranes, storing signals, glycosphingolipids acting as identity tags, mediating recognition, eicosanoids acting as potent local signals controlling inflammation, clotting, breathing.
They're everywhere, doing critical jobs.
Absolutely. And understanding them isn't just academic. It directly impacts medicine. Treating RDS in infants, using aspirin for heart disease, developing enzyme replacement for sphingolipidoses, designing asthma drugs. It all comes back to understanding these pathways. Every detail matters.
They really are the molecular language of health and disease.
Couldn't have said it better myself.
So here's a final thought to leave you with. We've seen how interconnected these pathways are, how inhibiting one can affect another, how a single enzyme defect can cause devastating disease. Given this complexity, what other unforeseen connections, maybe therapeutic opportunities, might still be hiding just beneath the surface? What bridges between seemingly unrelated conditions might we discover by looking even deeper into the world of these lipids?
That's a great question. The possibilities are likely vast.
Something to ponder. That's our deep dive for today. We really hope you feel much more well-informed, maybe even a bit awed, by the sheer elegance and critical importance of these molecular machines.